Go ahead.
Thank you for the introduction. In the next few minutes, I would like to show some new results of our research of a novel molecular mechanism behind the renoprotective effect of dapagliflozin. It is well known that tubular hypoxia has recently been rediscovered as a major factor in the progression of diabetic kidney disease. Hypoxia and O-GlcNAcylation are in a strong association with elevated glucose consumption. Previously, we showed that elevated O-GlcNAcylation could lead to kidney fibrosis. So clinical data support that SGLT2 inhibitors have renoprotective effect, but the exact molecular mechanism of action is still not fully clarified. So here we have investigated whether SGLT2's renoprotection is mediated part by the decrement of tubular hypoxia and O-GlcNAcylation. So briefly the method. We used a type 1 diabetic animal model in our experiment. We induced diabetes with a single intraperitoneal injection of streptozotocin in adult male Wistar rat. After the onset of diabetes, we immediately start to treat the animals with dapagliflozin for 6 weeks. So firstly, I'd like to introduce the metabolic parameters. Metabolic parameters confirm the development of diabetes, and dapagliflozin improve all of these functions. Renal parameters and the biomarkers of renal tubular damage also confirm the development of diabetic kidney disease. So we can say that in this type 1 diabetic animal model, dapagliflozin improved the renal function. Beside the renal function, we evaluated the structural changes in the kidneys by conventional histological stainings. So first, in this figure, you can see that the mesenchymal matrix expansion was significantly decreased by dapagliflozin, and tubular interstitial fibrosis was also ameliorated by dapa. After that, we determined the profibrotic processes. TGF-beta, CTGF, PDGFB, and alpha-SMA level increased in diabetes, and both parameters were decreased by DAPA except TGF-beta. So we can say that dapagliflozin have an antifibrotic effect. Inflammation have a crucial role in the development of diabetic kidney disease, so we investigated the pro-inflammatory cytokines in the kidneys. TNF-alpha, IL-6, and IL-1-beta expression level increased in diabetes, and dapagliflozin significantly decreased the amount of these pro-inflammatory cytokines. So we can say that DAPA has an anti-inflammatory effect. To investigate hypoxia, we use AGF-alpha, which is the biomarker of hypoxia. We measured the amount of AGF-alpha protein level in the diabetic kidney and also in vitro human proximal tuberous cells. So in both experiments, the AGF-alpha level decreased in diabetes and in high glucose treatment, and DAPA treatment prevents hyperglycemia in this tubular hypoxia. Finally, we measured the total protein O-GlcNAcylation and the enzymes which is catalyze the O-GlcNAcylation process. This figure shows that total protein O-GlcNAcylation is elevated in the kidneys in diabetes, and dapagliflozin significantly decreased the amount of total protein O-GlcNAcylation and the OGT enzyme. So, in conclusion, we can say that our results support that DAPA is an effective and safe treatment in experimental type 1 diabetic animal model. DAPA reduces tubular hypoxia and decreases O-GlcNAcylation, which is the novel mechanism of action of SGLT2 inhibitors in the kidney. So we hypothesize that these previously unknown mechanisms could be behind the antifibrotic and renoprotective effect of SGLT2 inhibitors. So thank you for your attention.